Welcome back to The Explainer. Today, we're zeroing in on a true microscopic mastermind. We are going to decode a highly adaptable biological villain that honestly is probably much closer to you right now than you think. So, let's jump right into this fascinating and, kind of terrifying world. 30%. Right now, as you're sitting there, about 30% of you are actually harboring this exact bacteria. Usually, it's just chilling quietly in the anterior of your nose, or maybe in areas of elevated moisture, completely harmlessly. But make no mistake, it's literally just waiting for a breach in your immune defenses to pounce. Now, the species name, aureus, actually comes from the Latin word for gold, aurum, because when scientists grow this bug in the lab, it develops these distinctly beautiful golden-colored colonies. But hey, don't let the pretty color fool you.
Part 1. Meet the golden bacteria.
Part 2. A recipe for food poisoning.
So this golden bacteria, it has quite the rap sheet. Let's kick things off with its baseline, sort of minor crime, turning a perfectly good meal into a totally miserable night of severe stomach upset. It's the classic recipe for Staph food poisoning. Typically, it starts with a food handler who's got the bacteria on their hands prepping something like processed meats or cheeses. Then, two things gotta happen. First, the food sits out at room temperature, which gives the bug time to multiply. Second, the previous cooking has already wiped out the competitor bacteria. You see, Staph doesn't actually compete very well with other organisms, so it really needs that empty playing field. Once it has it, boom, it pumps out massive amounts of Staph enterotoxins, or SEs. These are super antigens, but luckily for us, they don't get absorbed into our bloodstream from the gut. Instead, they stimulate the vagus nerve. Our body treats these toxins like a 5-alarm fire. Much like an ER doc using a stomach pump for a drug overdose, your vagus nerve triggers a natural stomach pump by inducing severe nausea and vomiting to just forcibly expel those super antigens.
But what happens if those same superantigens aren't sitting in your stomach, but instead they're growing completely unchecked in a walled-off area inside your body? A pus pocket. In this rare scenario, those toxins freely enter your circulation. The result is a devastating, severe systemic reaction that we know as toxic shock syndrome.
Part 3. The bacterial arsenal.
Now, when Staph actually breaks past our screen, it doesn't just wander aimlessly. It goes to war. This right here is its biological heist playbook. Staph possesses a truly terrifying array of virulence factors, specifically designed to attach, destroy, feed, and hide. Let's break down these specialized molecular weapons one by one.
First up on the list, adhesins. Think of these as molecular grappling hooks extending right out from the bacteria cell wall. Our tissues are held together by this complex web of proteins called the extracellular matrix, containing stuff like collagen and fibrinogen. While Staph uses these adhesin grappling hooks to bind super tightly to those structures, especially fibrinogen, and once it latches on, it's established a highly dangerous foothold right next to our cells.
But just hanging on isn't enough. The bacteria's gotta eat. Enter hemolycins. These are toxins, alpha, beta, gamma, and delta, that literally blast apart our red blood cells in human tissue. It's brutal. This rapid destruction releases a flood of vital nutrients that the bacteria just gorges on to grow at lightning speed. And as it grows and wreaks havoc, it attracts a massive swarm of our immune system's first responders, the neutrophils. As these immune cells fight and die off in the absolute chaos, they leave behind copious amounts of pus.
Part 4. Disarming the immune system.
Naturally, our immune system doesn't just sit back and let this happen. It mounts a really vigorous defense. But Staph is sneaky, and it's evolved some brilliant countermeasures to shut down the body's internal cops. A huge part of this is how it hides. Staph aureus produces an enzyme called coagulase, which it uses to clot the blood plasma in the surrounding tissue. It literally builds a protective shield of clotted plasma around itself, making it almost impossible for our immune macrophages to engulf it. This trick is actually so specific that clinical labs use it for identification. If a sample clots plasma, meaning it's coagulase positive, the lab absolutely knows they're dealing with the dangerous Staph aureus. If it's negative, well, it's usually just some harmless skin contaminant.
Okay, to really grasp Staph's most clever biological trick, we need a super quick anatomy lesson on our own immune system. Our antibodies are shaped like the letter Y. The top parts of the Y are the variables that grab onto specific invaders. The bottom stem, or the tail of the Y, is called the Fc portion. This tail is the constant end that signals the rest of the immune system to swoop in and destroy whatever the Y has grabbed hold of.
And right here is where Staph aureus shows its absolute dark genius. It produces something called protein A. Protein A grabs our antibodies by that constant tail, the Fc portion.
It literally attaches to the antibody backward. By doing this, it completely neutralizes our primary defense mechanism. It severely mitigates the activity of complement proteins and neutrophils. Basically, it blindfolds our immune system.
Oh, but wait, there's another evasion tactic. Unlike some other common bacteria, say group A strep, Staph aureus can actually pull off an inside job. It forces its way right inside accommodating human cells, like the ones in our skin, bones, and kidneys. By metabolizing while hidden deep inside our very own cells, it becomes far less of a target for our immune system. And this is exactly why some patients suffer from those incredibly stubborn, recurrent staph infections.
Part 5. The Rise of MRSA. Which brings us to a much more serious chapter in our explainer. Because as modern medicine developed new weapons against staph, the bacteria fought right back, kicking off a dramatic evolutionary arms race. Let's trace this epic back and forth. Back in the 1940s, penicillin arrived on the scene. Its core is this square, 4-carbon structure called a beta-lactam ring. And man, it worked beautifully. Until that is, staph figured out how to produce an enzyme called beta-lactamase to destroy that ring. So in the 1960s, scientists counterattacked. They developed methicillin by adding methyl groups around the beta-lactam ring to essentially hide it from the bacterial enzyme. We won the battle again. But then, the bacteria completely changed the rules, leading to a terrifying new emergence around 1999. The bacteria managed to mutate a critical enzyme used in its own cell wall construction. This new enzyme completely ignored the methicillin drug. It wasn't just destroying the antibiotic anymore. It had altered itself so it wouldn't even recognize it. And that gave birth to MRSA, methicillin-resistant Staphylococcus aureus. Because of this brilliant mechanism, MRSA is resistant to all antibiotics that have that beta-lactam ring, including all penicillin and cephalosporin drugs.
Now, for decades, MRSA was strictly a hospital-acquired threat. It persisted on environmental surfaces, basically preying on vulnerable patients undergoing treatment. But remember that 1999 date we just mentioned? That is when community-acquired MRSA, or CAMRSA, emerged. Instead of just striking the sick in hospitals, this completely new strain started infecting perfectly healthy people right out in the community, causing these rapidly erupting skin lesions.
The way this presented was both fascinating and highly alarming. These initial community-acquired lesions didn't look like your classic boils. They were small, discrete eruptions that spread extremely fast. In fact, patients were continually showing up at health clinics complaining of a mysterious spider bite, even though, no spider was ever actually seen. The spider bite was, in reality, a fast-moving CAMRSA infection.
So how does it build into such a rapid infection? Well, CAMRSA has elevated virulence largely thanks to a highly specific weapon called pantin-valentine leukocidin, or PVL. And here's a totally wild twist. This toxin is actually produced by a virus that has infected the staph bacteria itself. The PVL toxin consists of proteins that join together to literally punch deadly holes in the membranes of our neutrophils and macrophages. It actively destroys the very innate immune cells we send to fight it. Talk about an incredible defense mechanism. So how exactly did a bacteria get so good at this? It all comes down to its evolutionary origins. Staph aureus is, at its core, an exceptional genetic thief. Genomic analysis actually shows it's likely related to a completely harmless soil bacterium called Bacillus subtilis. But over eons, it just absorbed DNA and genes from other bacteria, and even viruses, as we just saw with PVL. That genetic theft is what drove its evolution from a harmless dirt dweller into a highly formidable human pathogen. We've seen it steal genes, hide from our immune system, destroy our cellular defenses, and literally outsmart our greatest antibiotics. This highly adaptable genetic mastermind is constantly evolving. Which leaves us with one final, very provocative question to consider. What will Staphylococcus aureus learn to resist next? Thank you so much for joining me for this explainer, and keep that curiosity alive.